It's my great pleasure to invite our first keynote speaker, Shyam Prabhakar, who will give a talk on the Asian Immune Diversity Atlas and the HCA Genetic Diversity Network. Shyam obtained his BTech in Electronic Engineering from IIT Madras and a PhD in Applied Physics from Stanford University. He was a sole recipient of the 2001 American Physical Society PhD Thesis Award of Beam Physics. Following postdoctoral fellowship in mathematics at Stanford and genomics at the Lawrence Berkeley National Laboratory, he joined the Genome Institute of Singapore, GIS. His lab uses single-cell assays, spatial omics, and novel algorithms for basic and translational research. He leads or co-leads several multinational initiatives and include the IDA project and also Atlas of the Cell States of Colorectal Cancer. He's also on the steering committee of the HCA Asia and co-lead the HCA Genetic Diversity Network. He manages GIS spatial and single-cell genomic platform, heads single-cell omics within Singapore National Precision Medicine Program, and is currently Associate Director of the Spatial and Single-Cell Systems and Senior Group Leader in Systems Biology and Data Analysis at GIS. Please join me in welcoming our first keynote speaker, Shyam Prabhakar.
Thank you very much, Ponpan, for the kind introduction. Really want to thank Ponpan, Varadon, Natini, Ananya, all the organizers, local organizers, for putting together this amazing event. First HCA Asia meeting in Thailand, and beautiful hotel. Everything's flawless, so we might just ask you guys to organize every one of these from now on. So, if you do too good a job. So, all right. It's my pleasure today to talk about the HCA Genetic Diversity Network and also IDA. So I'll be speaking on behalf of both of these organizations or networks, you can say, today. So first, come to the mission of the Human Cell Atlas, which Jay introduced very nicely, and that's to create a comprehensive reference map of the types and properties of human cells, right? So basically cartography, and then understanding the biology of those cells. I've underlined the word comprehensive reference map for a reason, right? And it's very relevant for us. So current maps, pre-HCA, okay, genomic atlases are not very comprehensive. They are predominantly derived from donors of European ancestry. So I'm showing examples of GWAS studies in the NHGRI GWAS catalog, the GTEx expression QTL consortium, NOMAD genome and exome sequencing. So you can see in all these cases, there's a huge overrepresentation of individuals of European ancestry. And so let's say, if you look outside the HCA, the atlases are not very comprehensive, okay? And it's not because they didn't try. It's not because they didn't realize that it's important to have diversity, right? So here you see in 2008, and I'm sure even before that, people were writing prominent review papers, geneticists were writing prominent review papers saying, we need more diversity in our studies, right? It's not enough to just profile individuals of one ancestry or one geography or so on. And so it was widely appreciated for scientific reasons, medical reasons, and so on. But so this paper was 2008. In 2011, 86% of the individuals in GWAS studies were of European ancestry. 10 years later, in 2021, 86% of individuals were of European ancestry. The needle had not moved, right? And I like to show this just as a sobering data point or factoid to say, we need to do just recognizing that you need to do something is not enough. There are many factors that stand in the way of diversifying our science, right? Primary among them, I think, is funding. So if all of the research funding, or most of the research funding, comes from very specific countries, right, in this field, then naturally, they're mostly going to fund studies within their own borders, and so they're going to profile the kinds of people who live in their country, right? So that is, but here I'd like to give a shout out to CZI, Chan Zuckerberg, based in the US. They have been amazing in diversifying their funding and funding people outside the US, all over the world actually. And I think that kind of initiative, that kind of funding strategy is very, very important for diversifying our atlases, okay? There are many other factors, don't have time to get into all of them. Cultural factors, many cultures, many population groups, they don't actually trust medical science, they don't trust genetics research because they've had bad experiences with it. Then there's simply resources, equipment, trained personnel are unequally distributed across the world. There are actually, say, religious reasons why certain people would hesitate to participate in this kind of research for various reasons. So all of that has to be tackled, right, if you want to diversify. But it is important that we do this because, as you know, disease incidence, disease mortality, it varies strongly across populations. Here I'm giving examples of diabetes, coronary artery disease, varies quite dramatically with where you live, what your ancestry is, and so on. Your risk of contracting the disease, severity of the disease, mortality, and so on. So these are two examples. There are many more. Broadly, your genotype, okay, the genetic variants you have in individual loci, your ancestry, which is, you can say your genome-wide genotype, your sex, your geography, all of those things influence your health, okay? So for example, population-specific disease risk. So here's an example for missense variant in African Americans, and then it's associated with underdiagnosis of heart failure in that population group. Your ancestry can affect your drug metabolism, and so the dosage of drugs ideally could be different in people of different ancestry. Whether you're male or female can affect your drug response quite profoundly. Where you live certainly affects your disease susceptibility or disease incidence. Infectious disease in certain parts of the world, cancers and diabetes and metabolic disease more broadly in developed countries. So mostly, so we know biologically, right, that all of these axes of diversity, genotype, ancestry, sex, geography, others as well, they are medically important, but we don't know precisely what their influence is, right? And so that has not translated so much into healthcare. And that's part of our job, to characterize human diversity to support precision medicine. Right? And if we don't do that, then we'll have strong healthcare disparities. Right? Our healthcare will be tailored to people, certain kinds of people living in certain parts of the world, and that's certainly not what we want. So how are we doing in the Human Cell Atlas? In terms of male, female, not bad. So this is Kian Hong from my group, analyzed PBMC data from 1,800 healthy donors, and there's some metadata available for them in the DCP. Roughly, slightly more than half of them are female, but reasonably balanced. That's great. If you look at ancestry and geography, okay, So this figure is slightly confounded. Some of the labels are ancestry labels, some of them are geography labels. But still you can see, okay, there is strong bias, 65% European. It's less biased than, say, the GWAS data set I just showed you, or the GWAS database I just showed you, which is 86% European ancestry. This also, actually, so I want to show this part, okay? If you take this out, this is actually IDA, which I'll talk about in the second half of my talk. If you take out IDA, you're back to the statistics of GWAS studies. It'll be around 86% European ancestry, okay? And that's because those are that's the typical cohort that you can recruit in the West, right? So if you want to make a dent in this diversity bias, right? If you want to move the needle, you really have to do what CZI is doing and what hopefully many other funders are going to do as well. Fund this kind of research all over the world, right? And these data sets come from CZI funding. Other groups, other funders are also getting into the act. And so that's the exciting thing. And funding research all over the world. Gates Foundation has done this for a long time for infectious disease. And that's the kind of model we need for the Human Cell Atlas. So within the HCA, the job of making this happen falls on the shoulders of the Genetic Diversity Bio Network, which I co-lead with Jimmy Yi and Sarah Tishkoff. And it's exciting, I would say it's an exciting responsibility to have because diversity is a scientific frontier. As scientists, you want to jump into fields where you don't know much, right? Where very little is known. This is a field where almost nothing is known. Okay, even the most basic questions have not really been addressed comprehensively. So, highly recommend. And HCA Asia is the group that can get into this field and make a mark in this field. So, what does a GDN want to do? Our mission is we want to characterize the impact of genes and environment on molecular traits in cells from people all over the world, diverse people all over the world. We want to narrow that down to a few axes of human diversity. We have to prioritize. We have to start somewhere. So as a fruit of many discussions, I think we had 5 international roadmap discussions within GDN, brainstorming on what we should do, how we should do it. And the consensus was that we should understand how genetics, ancestry, sex, age, geography, and lifestyle influence our cells, influence our tissues, influence our biology. And we want to do it paying particular heed to ethics principles, which are not the same all over the world, right? So when you do diverse research in many cultures, many communities, many countries, the ethics issues are very, very different in each place, right? So you have to take that seriously, put some investment in that. And we want to do it in an equitable manner, which is by equity you mean you want to represent, of course, individuals all over the world. You want participation, and Human Cell Atlas is a major vehicle for that, for participation of people all over the world in the research itself, not just as donors of samples, but as people performing the research. And largely the research has to benefit people all over the world, right? It should not benefit just a handful of groups. So that's the equity angle, or the equity goals for the GDN. And ultimately we want to do all this so that we support precision medicine, in our case in Asia, but for GDN all over the world, right? So what are we going to do specifically? We want to generate or diversify the Human Cell Atlas, do single cell analysis, looking at all of these axes, and also for the look at the genetics of our cell types in each population. So the QTLs, the quantitative trait loci, may not be different in, say, let's say if you look at individuals in Thailand. Thailand has many population groups that Varadam and Ponpan are studying. But let's say Thai individuals as one group, right? If you look at the genetic effect of one variant over here, is it the same as a genetic effect of that variant in a European ancestry population? Is it the same as the effect of that variant in an Indian ancestry population, for example, right? Very basic scientific question. We don't really know the answer. Right? There have been some comparative EQTL studies, but those in different populations, but those studies have been done using bulk gene expression analysis. And bulk gene expression analysis is very limited. It just tells you the average properties of of cell types. If there's a very specific cell subtype or cell state that's responding to this genetic variant differently, say, in Thailand versus in San Francisco, okay? And it's quite likely, right? Over here you have different climate, different genetic background, different environmental influences, different lifestyle, different infectious disease burden. So it's quite likely, right, that your cells will respond differently. And you really need single cell analysis to do that. Right? So QTL analysis will be a big part of the HCA GDN. And then methodologically, we're trying to get everyone to harmonize how they generate data, how they collect metadata, and how they represent and share the data. Then it won't be just individual studies. It'll be, we can do meta-analyses across many different studies in the GDN. And of course, we need new algorithms. So there are lots of algorithms for saying what cell types exist in this sample. What is the trajectory of this cell type as it differentiates? What are differentially expressed genes between case and control, disease and healthy, right? There are not so many algorithms for addressing the scientific questions that the GDN wants to address. And so we do need to develop new methodology. And then finally, we want to take all these healthy atlases and then in our own local communities, use them as references for disease studies. So I hope, so we already have quite a few members in the GDN, and they've submitted their projects in that spreadsheet. We hope to have more GDN projects as a result, and particularly in Asia. And Jay mentioned flagship projects that we're looking for new flagship projects in HCA Asia. We hope they'll contribute to the GDN and sign up for the GDN meta-analysis. If you want a more detailed outline of the motivations, rationale, methodology, structure, and so on of the GDN, take a look at the roadmap draft document that we put up online, and that's much more extensive. Okay, so in the Asian context, Human Cell Atlas Asia was formed, I think it's fair to say, before the HCA Genetic Diversity Network was formed. And so it's kind of coincidental, right? But as it turns out, HCA Asia is a fantastic vehicle for achieving the goals of the Genetic Diversity Network. Because 60% of the world's population lives in Asia. It's the biggest continent. The two world's most populous countries are here. We have tremendous genetic, ethnic, cultural, environmental, socioeconomic diversity. So if you want to understand human diversity, Asia is a fantastic place to test all of these ideas. And within HCA Asia, as Jay mentioned, 4 years ago, I guess, in Jeju, in the second Human Cell Atlas Asia annual meeting, we got together and we said, okay, let's turn all these principles into action. We know that Asia is underrepresented. We know that we want to contribute Asian samples, address Asian research priorities in the Human Cell Atlas. that all of those priorities were laid out in the first meeting that Jay and Piero organized. In the second meeting, we said, okay, before we leave Jeju, we want to have a plan for what is the project, what is the science we're going to do. And so IDA was a result of that discussion, started out with just Singapore, Japan, and South Korea. As you can see, it's expanded to 8 or 9 countries now at various stages of onboarding. And so this is really a flagship, and I hope it'll serve as a template for many more consortia within HCA Asia. So IDA decided to start with PBMCs, peripheral blood mononuclear cells. In a sense, the most convenient place to start. We can collect diverse data sets all over the world. You can freeze the samples, cryopreserve them, ship them if needed, or process them locally. And as it turns out, we were not alone, right? In the broader human cell atlas, PBMCs are the most common cell type. All right, so what's unique about IDA? It'll be, and I've listed the names. It's a huge consortium, okay? I've listed the names of mostly the PIs over here that are driving the respective IDA teams, but there's large groups working with them. So it's exciting to have so much so much interest in this consortium. One unique aspect of IDA is that we're doing 5' single-cell RNA-seq, which means that we'll get the transcription start site, we'll get enhancer RNAs in addition to gene expression levels. Also profiling chromatin state, we're genotyping all the samples, so any diversity-related project that you do, you will want to genotype the samples as some kind of independent measure of the ancestry and genetic state. And thanks to the 5' strategy, we also get the B cell receptor and T cell receptor diversity at single cell resolution. And it'll be very interesting to compare that in the healthy state, in inflamed state, disease states, and so on. And so IDA did start out asking exactly the same questions that I've laid out for the genetic diversity network in the Asian context. But I think it's kind of a case study or proof of principle for what we can do on a much larger scale globally. And it'll be very important for precision medicine in Asia and serving as a healthy baseline for disease omics. And that's why in Singapore, IDA Singapore collaborates with Helios. Helios is Singapore's national precision medicine cohort. And the exciting thing there is that so John Chambers is leading that cohort, and there's a lot of metadata that they're collecting on the IDA individuals. So if you're starting your own diversity-related HCA or HCA Asia project, it's a very good strategy, I think, to align yourself with your national precision medicine program, because then you can leverage all the whole genome sequencing that they're doing. You can leverage all the, say some of them have longitudinal cohorts. You can leverage all of the phenotyping that they're doing, which you may not easily have resources to do yourself. So we're very fortunate in Singapore to have that. And specifically in IDA, we're going to compare the healthy samples to pre-diabetic samples, type 2 diabetes samples, cancer blood, which is has strong diagnostic potential and actually could tell us something about the biology as well. So the data generation strategy we're using in IDA is increasingly common in the Human Cell Atlas now, and this is how you reduce batch effects and reduce costs. So if you want to do any large-scale single-cell project, there are many ways of pooling samples and then reducing the number of assays, single-cell assays you do. So basically pool cells from many different individuals and process them all in a single reaction for single-cell library generation. We started out using the method developed by Jimmy Yee. It's called demuxlet, which is you just pool the cells without doing anything, but they're cells from different individuals, so they'll have different genotypes, right? And then some of the single-cell RNA-seq reads that you have will cover those SNPs, and so you can get some genotype information from the single-cell RNA-seq reads from each cell, right? And any particular SNP will be covered at a very low coverage in general, but then if you take all of the single-cell RNA-seq reads from one cell, you're covering a lot of SNPs, and a lot of those SNPs are ancestry informative. Right? So then bioinformatically, you can very easily demultiplex these pooled cells and say, and really pretty accurately say this cell came from this individual, this cell came from this individual, and so on. And that's a demarks-led strategy. You don't need to do anything extra to the samples. You don't need to add any antibodies to them. You don't need to add any other peptides or oligos or anything like that. And so it's an easy workflow, and we're using this on a very large scale. And then bioinformatically over the years, we've developed a fairly large, long, complex data analysis protocol. So again, if you're starting down this road, you should know that the data analysis, so setting up the consortium will take you a huge amount of time. So getting the IRBs, getting access to the samples, recruiting donors, doing pilot studies to see if your assay is working and so on, that takes a huge amount of time. The actual data generation doesn't take very long. And then the data analysis takes an even longer time, right? Because whenever you have a data set that's so large and so complex. So then if it's such a big task, it deserves a careful pipeline, right? And what's unique about our pipeline is that it's two-stage. We do the QC, cluster the cells at a very coarse level. So loose QC, coarse cell type clustering to identify major cell types. Okay, let's say cell lineages, T cells, B cells, myeloid cells, things like that. So very low level clustering. And then we break apart the data set. We take the T cells separately, we take the B cells separately, and for each lineage or each partition of the data, we start all over again. We say, now let's do QC on the T cells, then let's recluster the T cells to identify subtypes within the T cells. Let's do a separate QC on the myeloid cells, because myeloid cells will have different QC properties. And then subcluster just the myeloid cells, and when you do that, you get better feature selection, right? So when you subcluster the myeloid cells alone, you're only using the genes that are informative about differences between myeloid cell subtypes. You've thrown away all the T cell markers, you've thrown away all the B cell markers that are irrelevant, and so then they have those markers no longer add noise to your myeloid cell clustering, right? And so then you get much more accurate clustering, and of course the fact that you've done tighter QC that's myeloid specific, again helps you cluster the myeloid cells. And then you have, so this two-stage pipeline we're using, I'll show you an example and hope to convince you that it's working very well. Very happy to say it's been adopted by the Immune Cell Atlas, the HCA Immune Cell Atlas team as well, and hope many other teams will adopt it. So this is what I mean by two-stage QC. All the gray dots, so this is QC cells in QC space, okay? X-axis is the sensitivity of your single-cell RNA-seq assay, how many genes you're detecting in that cell. Y-axis is the mitochondrial read percentage, which is an indicator of how damaged your cell is, okay? So you want your cell to be low down, low on the y-axis, and far to the right-hand side on the x-axis. Okay? So the gray dots are the output of our loose QC, all the cells that pass loose QC. The black dots are the cells that pass the stringent cell type specific QC. And you can see that for each of these major lineages or major cell types, the distribution of cells in QC space is different. So your QC cutoffs have to be different, right? And using this elaborate procedure, we finally got this beautiful distribution of cells in gene expression space. Discarding way more cells than people normally discard, but you see the benefit. If you looked at PBMC single-cell RNA-seq UMAPs, you'll notice that you see the T cells on the right-hand side, okay? The T cells and the NK cells, the blue and the orange. You'll notice that they have a lot of structure. It's not just one sausage-like blob, right? And that structure means that you're very crisply identifying the subtypes of T cells and subtypes of NK cells. If we had not done this elaborate two-stage QC, cell types QC, and also I would say there's a testament to the high data quality in IDA. The DMUX-led pooling samples to reduce batch effects, that also helped the clustering. And then simply iterating on the assay to make sure that the viability of the cells was good. The fruit of all of that is this beautiful UMAP with well-resolved cell types. Okay. Now on the right-hand side, you can see we're also profiling TCR, BCR. So the red dots mean that we got some TCR data from the cells. So it makes sense that the T cells have red dots because you get TCR from there. The B cells, plasmablasts, have blue dots on the right. So you got B cell receptor information from there. So these are the major lineages, and then we'll pull each of these islands apart, and then we'll subcluster them to identify subtypes. Now, excitingly, already with the phase 1 IDA dataset, which is 3 countries, South Korea, Japan, and Singapore contributing 3 ancestries, okay? So Southern Chinese, it says Malaysian, but it's actually Malay. They're not Malaysian, they're in Singapore, and Indian ancestry, right? So we've got data from 5 ancestries from 3 countries in phase 1 of IDA. And you can see here and it's overlaid on 1,000 genomes PCA plot, just the Asian samples from 1,000 genomes. So you can see here that the IDA samples very nicely cover the diversity of Asia, the genetic diversity of Asia. Not comprehensive, obviously, but broadly span the major axes of genetic and diversity in Asia. So we can have a lot of interesting science out of this, right? We're just getting started on what we can learn from here. So one is, are we seeing differences between ancestries? Okay. Now here, this is Indian versus Chinese in Singapore, relative abundance in gene expression space. Okay. So orange means that, say orange at the top, means that those, let's say here, okay. So these B cells come more from Indian donors than from Chinese donors after you normalize for all the depth of sequencing and things like that, right? This T cell subtype is less abundant in Indian donors compared to Chinese, Indian ancestry donors compared to Chinese ancestry donors. Same for Malay ancestry versus Chinese ancestry analysis, right? And this is the beauty of doing this using single cells. If you did bulk analysis, all of these cells would be grouped together as one cell type. And the orange and the blue would average out, and you would say, okay, Indian ancestry and Chinese ancestry individuals have the same abundance of that cell type. But here you're doing it's more unbiased. You can essentially define arbitrary subtypes, sub-subtypes, and sub-sub-subtypes in gene expression space and say, essentially, gene expression tells you the distribution of cell phenotypes, and you can see for every single cell phenotype that I'm seeing in the blood. Does people of this ancestry have more of it? Or do people of that ancestry have less of it? Right? So that's a cellular resolution analysis. We're also doing the more traditional, cell type resolution analysis. So you can see, and we're looking at, as I promised, ancestry, age, and sex. So the first example is ancestry. So you can see here that Chinese ancestry individuals versus Indian ancestry individuals in Singapore. So Chinese ancestry individuals seem to have more NK cells. And these are all processed side by side, collected side by side, batch randomized. So there's no batch effect here. This is consistent across many, many individuals and many, many batches. Then effect of age, plasmacytoid dendritic cells. If you look closely, you can see a small negative trend. Older individuals seem to have a lower proportion of plasmacytoid dendritic cells in their blood consistently. And then if you look at plasmacytoid dendritic cells, again, male versus female, the effect of sex. You can see here that it's mostly balanced, but there are some imbalances, and some of these are not statistically significant. But some of the other cell types, we're actually seeing statistically significant differences between male and female. At this level of resolution, we can validate it using facts, right? But single cell analysis allows you to do ask this question, male, female, age, ancestry, at much higher resolution. And that's an ongoing task. So very, very broadly, right, what is IDA going to contribute? And we hope to wrap up this analysis within the next, okay, I'm not going to stick my neck out, within the next few months. So, but before too long. We want to look at these axes of human diversity. How do they affect, very specifically, right? How do they affect cell type proportions? How they affect subtype proportions? Cell state enrichment, as I showed in those orange and blue UMAPs. How do they inflect the expression of specific genes differentially expressed based on genotype, ancestry, age, and sex? What is the regulatory variation, meaning the transcription factors that drive these differences between these different population groups? And lastly, the genetics of gene expression, so quantitative trait loci affecting expression levels, splicing, transcription start site usage, and so on. And how does that vary across these population groups, right? Is this QTL stronger in females versus males, things like that. And then finally, IDA, we've already generated some of the data. IDA will contribute to diabetes, cancer in Asia. Also, enhancer RNAs and transcription start site usage. Because we're doing 5' single cell RNA-seq, and we're seeing enhancer RNAs, we can even treat that as a trait, as a molecular trait, instead of just gene expression levels. Enhancer RNAs tells you essentially the expression level of an enhancer, how active an enhancer is, right? So this is a goal, and we're well on our way, so watch this space. I want to thank the GIS IDA team. Kian Hong is over here in the audience, who made most of these slides. Shweta is a program manager for IDA. Lamin and Prasanna generated the for GIS IDA. Lamin and Prasanna generated the GIS IDA data with Deepa, who was there in the lab before, and Kian Hong. Zuye, Radhika, and Eliora, and with Kian Hong analyzing GIS IDA data. I'm very thrilled to have Jason with us now in GIS since January, joining the GIS IDA team. Also, the wider IDA team, there's many people to thank, and I have not even listed all of them, but I mentioned many of their names. I mentioned our funders also, CZI and A-Star. So, thank you.
Thank you, Shyam, for a wonderful talk and very brilliant talks that help kickstart the meeting really well. And we all feel excited for the next 2 days that we as a community really need to produce more data of the Asian population to fill the gap. Thank you. Is there any question from the floor?
Hi.
Please go to the microphone.
Hi.
Hi, Kirsten.
Hi. Kirsten Meyer from the Wellcome Sanger Institute. Really lovely overview, thank you. I was just wondering, within the ADA cohort, have you recorded when the samples were taken with respect to the COVID pandemic? Because it would be really interesting to integrate that with some of the other sort of COVID PBMCs we are collecting at Sanger.
Great question. The samples whose results I showed here were collected before the pandemic, so no effect of COVID. But now we have a new batch we're profiling during the samples collected during COVID. So that'll be very interesting. Obviously, we won't know perfectly everybody's disease state or disease history.
Yeah.
But we do have a separate project on COVID PBMC single cell RNA-seq analysis. So we're quite familiar with the COVID signatures, and we'll definitely be looking for them in the data from our next batch.
Thank you.
Hi, Shyam. Ishan from IT Delhi.
Hey, Shyam.
I was quite curious about the plot that you showed with the abundance ratios. So, is it at the level of single cells that you're saying something is more from a particular ethnicity, or at the level of a cluster that the false coloring is happening?
We're looking at it at all levels of resolution. So major cell type, subtype, cell neighborhood, expression neighborhood, everything.
Okay, so that color is not at the level, because...
Oh, so the colored UMAP you're talking about?
Yeah.
That's at the level of 500 cell neighborhoods in expression space.
Shyam, just a technical question. This is regarding pooling of the cells without actually adding barcodes. And you expect to be able to identify impacts on the expression of single SNPs. Unless the number of cells for each type or the depth is very high, that will be very hard. What you will probably be able to detect are haplotype effects. Would you agree with that?
Yeah, I think you're spot on the statistical power. We don't have the statistical power that GTEx has, because they're doing bulk RNA-seq, which is less noisy. We're doing essentially pseudo-bulk RNA-seq by pooling cells from one cell type. So for if we're just looking at, say, T-cell EQTLs, right? That's a pretty good pseudo-bulk. There's a lot of T cells from one sample. The data won't be so noisy. If you want to do a plasmacytoid dendritic cell expression QTL, we're pooling PDCs from one individual, that'll be a bit noisy. We'll have less statistical power. So that's definitely a factor.
No, but when you're pooling over various ethnicities or ancestries, in that case, the same SNP will not be represented in all ancestries. So you'll have to sort them out, and there the sample sizes even go down even further. On the other hand, if you're looking at a bunch of SNPs like haplotypes, you probably will be in a better shape. We can discuss that later.
Sure,
yes.
yeah. But I think rare variants will be population specific for sure. So what we can compare across ancestries is the common variants.
Hey, hello.
Hey, Swain.
So I'm Swain Chen. I'm at the Genome Institute of Singapore, also an Amazon Scholar with AWS. And so, Shyam, that's a great talk. Seen lots of your work before, and sort of glad to see the whole pipeline together. So I was also curious about this result of the differential abundance of cell types. And so maybe to take a slightly different angle on it, this idea that, as you said, you can sort of keep subclustering as much as you want. And when that starts to correlate with ancestry, right, then really, are you actually talking about different cell states? Maybe we can sort of talk about that, what your views are as to like how far effectively you should go down. And now you start seeing, well, QTLs, maybe you're starting to cluster QTLs, but it's really the same cell type. So you're going to talk about that issue, the interaction between ancestry, genotype, versus the definition of cell types or substates.
Exactly. So we want to go down as high, so let's say as high resolution as the data allow. And I think those visualizations are very useful. Those visualizations tell us what is the shape of the biological effect in gene expression space. And that's the advantage of single cell analysis. This is not published in our COVID study. We found something very similar, that we found that the effect of COVID, severity and longitudinal trajectory and so on, affects a very peculiar set of neighborhoods in gene expression space that isn't defined by any very specific cell type or any classical cell type, but is defined by specific gene expression markers and specific signaling pathways. And so I think that when you look at it in an unbiased way in gene expression space and you don't have any preconceptions about which level of resolution you should look at. So you look at as high resolution as the data permit, then you start to see patterns you did not see before.
Okay, thanks. That's super cool and great talk. Thank you.
Thank you.
Hello, Dr. Prabhakar. My name is Abhay. I'm from IIT Delhi. I had a question regarding QC and filtering. So in metabolically active cells, we have high mitochondrial content, but if we do a global QC, we would get rid of them. So, but if we don't do, then we are including dead cells. So how do you tackle that issue?
Yeah, it's always a trade-off in QC, and it's subjective. no machine learning algorithm is going to tell you where to set the QC cutoff. So what we do is trial and error. We'll eyeball the distribution, we say we'll draw the QC cutoffs here, then we'll take the cells that we threw away and say, do they constitute a specific cell state that we're losing? So we just do it empirically. And we try and, let's say you have a discrete metabolically active cell state with a high mitochondrial content that you've thrown away, then it should have some very specific gene expression markers. It should match some biology you know from the literature. And so if you see that, we say, oops, we made a mistake, let's not throw it away, let's bring it back. But if the cells that we throw away show nothing very interesting or any meaningful patterns, they only show signatures of damage, signatures of stress, signatures of low sensitivity, then we say, okay, fine, it was justified to throw them away.
Thank you.
So when you show biases in T cell, B cell, or NK cell subset corresponding to different ethnicities, have you checked to what extent these could be due to bias usages of particular TCR, BCR, or KIA genes, or what you're showing now is purely due to phenotypic characteristic regardless of specific TCR, BCR, or KIA genes?
Are you proposing that a different TCR clone, because this cell has a different TCR clone, it'll affect the transcriptome, the expression of the other genes?
Yeah, but then if you think about maybe like certain ethnicity would have a particular or like biases in like HLA haplotype, right? There could be some biases for certain TCR genes, maybe. And maybe this could drive, like different subset, but whether they're really subset, if we consider like different subset of TCR as subset, or should we try to remove TCR or BCR genes first and then just try to purely look at, like T cell subset as phenotypic characteristic regardless of TCR?
So the gene expression phenotypes I showed, they are not driven so much by the expression of TCR genes. Maybe I'm misunderstanding the question, but.
I mean, in certain contexts, especially maybe like in infection or certain cancer, sometimes we have like, strong clonal expansion of certain TCR clone,
yes,
and then that
got
could
it,
drive,
got it. Yeah, absolutely.
yeah, like a separate, like, cluster, right? So I'm just wondering, like, if this could happen even, like when you look at
very
like
much can.
ethnicities.
Very much can. So if I can broaden from, population-specific TCR diversity to population-specific XYZ, right? Population-specific infectious disease burden, population-specific diet, population-specific lifestyle, population-specific inflammatory allergies. It's like Singapore has a lot of allergy burden, right? All of that will shift your expression profile. It's not necessary that the genotypes of the SNPs are causing that shift in expression state. The environment could well be causing the shift in the expression state. So when we say it's ancestry-specific gene expression, we don't mean it's genetics-specific gene expression, okay? Because ancestry is confounded with ethnicity, and ethnicity is culture. Ancestry is confounded with geography, right? So ancestry is confounded with so many things. Ancestry is confounded with race. It's hard to, so ancestry, race, ethnicity, it's hard to separate them, tease them apart statistically in cohorts of this size. You need a massive cohort to say that this is a race effect and not an ancestry effect and things like that.
Thank you. We have time for last question.
Hello, this is Jingjing from Singapore Immunology Network. So I have a question about the cell type proportion. So did you take the number before or after filtering? Because it's a possibility that some of the cells that just age-related effect are just more fragile rather than the number decreasing.
Excellent question. We always worry about that. So we're looking at the proportions after filtering. But this is true for any science you do with single cells, not just our study, right? Any science you do with single cells where you're counting cell proportions, they could always be biased by which cells survive and which cells don't, confounded by which cells survive, which cells don't in your assay and in your QC. So what we really need to do is an independent validation assay, and we're working with SIGN on that, so with your colleagues. Yeah.
So thanks, Shyam, and thank you for all the fruitful discussion.